So now we are going to learn about the diastereotopic hydrogens. So the diastereotopic hydrogens here means we have to find the relation between the two hydrogens, and when we are going to replace the hydrogen with the deuterium on first reaction, in first reaction we are going to replace the hydrogen with deuterium, we are getting this particular product. And if we are replacing the hydrogen with deuterium in the second reaction, we are getting this product. So what's the result is, like, we have a hydrogen which is we have alkene as a reactant. So we have alkene and we have to find the relation between these two hydrogens. What is the relation between these two ligands? Hydrogen is also a ligand, hydrogen is also a ligand, methyl is also a ligand, hydroxide is also a ligand. And if we are going to calculate the relation between the two hydrogens, what we have to do is, we have one H on this end and another CH3 on this end. So we have a deuterium on this end and hydrogen on this end. So how are these molecules related to each other? Like, if we are going to replace the hydrogen with deuterium, we are getting this and this. These two are diastereomers. These two are diastereomers. If these two are diastereomers, then the relation between the hydrogens are also diastereotopic. So these two hydrogens are diastereotopic. So what's the relation between these two hydrogens is, these two hydrogens are diastereotopic. These two hydrogens are diastereotopic. It's a very important thing that you have to learn in the stereochemistry so that you can apply in the spectroscopy as well. So now consider the relation as an enantiotopic. So if we have two hydrogens, HA and HB, and if we are going to replace the HA with X and HB with X, so these two are enantiomers because they are non-superimposable mirror images. Because HA can reflect HB and X can reflect X. So these are non-superimposable mirror images, and if the final products after the replacement forms the non-superimposable mirror images, then we can say that these two hydrogens are enantiotopic. But you have to consider each and every example very carefully, because the relation here is enantiotopic because we have non-superimposable mirror images between these two compounds. Now the symmetry method, I am going to explain you about the symmetry method as well, but you have to apply the symmetry method in spectroscopy part. I am going to explain you the rules and the protocols of this particular symmetry methods, but you have to actually apply in the questions, like the approach to solve the questions to symmetry method. We are going to learn that particular thing in spectroscopy only. So if the ligands are related with CN, if we have a ligands and those ligands are related with the CN axis, I have considered this particular compound like this. This is a compound, we have a methyl group here and we have a methyl group here. I have marked this particular methyl group as A and this particular methyl group as B, and we have hydrogens HA and HB. Just consider a CN axis passing between this. We have a CN axis, just consider I am passing the C2 axis. If I am rotating the molecule with the C2 axis, I am getting this as a product. If I am rotating a molecule with the C2 axis from here, like I am rotating the molecule with 180 degrees, I am getting this product. If I am rotating a molecule with 180 degrees with a particular axis, with the CN axis, that is the C2 axis here, so I am getting the same product. And if I am getting the same product, it means this HA and HB are homotopic. HA and HB are homotopic because I am getting the same product. HA and HB are homotopic. CH3A and CH3B are also homotopic because they also considered as a ligands. So if they are also ligands, so that's why HA and HB are also homotopic, and CH3 and CH3B are also homotopic. So if the ligands are related with the CN axis, then the relation between the ligands will be homotopic. This is the first rule of symmetry method. Second rule of symmetry method is, if the ligands are reflected by the plane, then the relation between the ligands is enantiotopic. Consider this particular example. We have CH2 and CH3. If I'm going to pass a plane like this, in which this particular CH2 and CH3 aldehyde group and the methyl groups is bisecting, but HA and HB are reflecting each other. HA and HB are reflecting each other. They are reflecting each other, and if these particular hydrogens are reflecting each other, so the relation between these two groups is enantiotopic. So HA and HB are enantiotopic because we are considering the plane of symmetry. If the plane of symmetry is there in the molecule, and the two hydrogens are reflecting each other, then those hydrogens are enantiotopic. So the relation between these two hydrogen is enantiotopic. This is the second rule of symmetry method. The third rule of symmetry method is when the ligands are related with the center of symmetry, that is the inversion center. Center of symmetry, I have told you about the center of symmetry. It is also known as the inversion center. So when the ligands are related with the center of symmetry, then the relation between the ligands is enantiotopic. How I am saying this? Just consider this example in which I have marked inversion center here, COH here, COH on this side. Inversion center. For inversion center, we have to go opposite direction in 180 degrees, and the 180 degrees, and in opposite directions we have to go in opposite direction 180 degrees. The environment on both then should be same, only then the inversion center is going to exist. So COHB and COHA reflecting each other. CH3b and CH3a are also opposite to each other. OHA and OHB are also opposite to each other. It means there is a COS exists. So there is an inversion center existing in this particular compound. So there is I, that is there is I which is existing in this particular scaffold. So we can say that OHA and OHB are enantiotopic. We can say that OHA and OHB are enantiotopic. We can say COHA and COHB are enantiotopic. We can say this as well. CH3A and CH3B are also enantiotopic. So these are the ligands. If the ligands are related with the center of symmetry, then the relations between those ligands are enantiotopic. So here the particular I is existing in the molecule, so OHA and OHB are enantiotopic, COHB and COHA are enantiotopic, CH3B and CH3 are enantiotopic. So in such a way you can relate the relation between the ligands. Now the last rule for the symmetry method is if the ligands are bisected by the plane, then the relation is diastereomers, because just consider this example. So this is an example in which we are going to relate through the bisecting plane. So we should have a bisecting plane. We can draw this structure as this as well, because Cl and H on this side and Cl and H on this side, and opposite we have HA and HB, and we have HH on this side as well. So we have to bisect a plane. So if we are saying that if the plane is passing from C1 and C2 carbon, then these hydrogens are reflecting each other, and if these hydrogens are bisecting, it means they are diastereotopic. What I am going to say is, consider this structure of cyclobutane. We have a chlorine here, we have hydrogen here, we have hydrogen HA and HB on this particular side, and we have hydrogens here. I have to tell the relation between HA and HB. So plane of symmetry, particularly bisecting plane of symmetry, if there is bisecting plane of symmetry, those bisecting hydrogens are known as diastereotopic. So if we are going to calculate the relation between HA and HB, we have to see the bisecting plane. So if I am saying, if I am going to pass a plane like this, just imagine this ring like this particular carbon. First carbon is front of you and second carbon is also in front of you, and this a ring like this. This is a ring like this particular thing. And if you are going to pass a plane, and if you are going to pass this particular plane, H and HB are bisecting each other. And if H and HB are bisecting each other, and these two hydrogens are reflecting each other, you can also consider the plane like this as well. If you have a Cl here and H here, HA and HB on this particular side, and we have H and H on this, H and H on this, then also H and HB are diastereotopic. Consider this particular example, ClH, Ha and Hb. If we are going to pass a plane from C1 and C2 and the Ha, Hb, chlorine and hydrogen are bisecting, then the Ha and Hb have a relation of diastereotopic. There is another example which we can do. There is another small short trick you can say, or a conclusion that we all have made by learning the stereochemistry from different standard books. So we are going to come a conclusion. If we have a structure like this and if we have a chiral center, suppose we have OH on this side and OH on this side as well, we have two OH groups and this is a chiral center because we have a chlorine and a bromine. So the ligands which are adjacent to the chiral center are also diastereotopic. They are diastereotopic because the ligands which are adjacent to the chiral center are diastereotopic. You have to learn this particular thing because it's very important point. The importance of this symmetry method you are going to learn in the spectroscopy chapter. So the ligands either OH, either H or either CH3 is there, so that are diastereotopic. So just consider this example. We have H on this side, we have OH, we have chlorine and we have methyl. So this is a chiral center. If this is a chiral center, these two hydrogens are diastereotopic, and if these two hydrogens are diastereotopic, we are getting the two different signals in the spectroscopy for these two hydrogens.